You guys have already covered that. So that's how it gets from out here to in there. There's some question marks, right? But that's our best approximation of how it happens. Okay, let's talk about steroid hormones. All right, so you guys have heard about steroids, right? Hormones that influence a variety of physiological processes in us. And you should have some appreciation that they're signaling molecules of some sort. Right? They circulate and they signal. So this is cholesterol. Very hydrophobic, right? It has this little hydroxyl here, which can be a hydrogen bond donor and acceptor. That's about it. And the rest of this is all hydrophobic. All right, so the function of cholesterol is twofold. It makes the membrane less fluid, and it makes the membrane more fluid. You guys confused? Yeah. Makes it less fluid and it makes it more fluid. Let's talk about a membrane just real quick. So we have our bilayer. This is the hydrophobic core, and this is our hydrophilic region where the head groups are. Because of this, the shape of cholesterol, just kind of approximate it. Fat up here, skinny down here. So what it does is it increases the density here. So in this zone, we'll call this the interface zone. It increases the molecular density. Because it's fat up there. Makes this region more stable, less fluid. But then down here it's skinny, and it makes this region more fluid. All right? So it has an overall stabilizing effect on the membrane while increasing its fluidity, its fluid properties. It's kind of weird. That's all based on its shape. So that's its function. It's very important in animal cells. All eukaryotic cells, actually, make steroids. And this actually, the cool thing about cholesterol is it does this at a variety of temperatures because of its shape. So our membranes can actually be fluid at a number of temperatures without changing the number of double bonds, cis double bonds in our fatty acids with cholesterol, because it does this weird thing. It makes this part less fluid, this part more fluid. Okay, so cholesterol, really good at membranes, terrible signaling molecule. Right? Why is it a terrible signaling molecule? Too hydrophobic. It wants to be in a membrane. All right, so if we want to make this into a signaling molecule, and we do, we're going to have to do something. What would be the easiest place to get rid of some hydrophobicity? If you're an enzyme, you're like, oh, in one step I want to make this very much more hydrophilic. Where would you cut something? Maybe right here. Get rid of that thing, whatever that is. Because all this is like this cyclic stuff. I don't even know about the chemistry there. That's too hard. We can do this. This would be the first place where we can make some changes and make this thing more hydrophilic. All right, so if there's a mixed function oxidase, we know how these work. NADPH, NADP plus. This one takes 2 molecules of oxygen, converts to 2 molecules of water. You might be asking where the other 2 oxygens go. They're going to end up right here. So I'm just going to draw this little piece right here. Save my arms some work. And the rest of the cholesterol is down here. So we've oxidized these 2 carbons right there, and that sets us up to do a cleavage event. There's something called a desmolase. And your book has a slight typo here. A desmolase is kind of like a mixed function oxidase. Except in this case, the electrons are going to end up right here to resolve a carbocation. Similar chemistry we've seen before. So in your book, they say it takes 1 molecular oxygen and converts it to 1 molecular water. It actually converts it to 2 molecular water. Don't be fooled. The book has a little typo there. All right, what's happening here is we're breaking this bond and moving it up there. And then we're using reducing power to resolve the resulting carbocation that would be there. All right, so then what we get something looks like this, and then the rest of the cholesterol is down here, right? And we end up breaking off this little thing. Yeah. All right, and this is going to be metabolized. We use it for energy. And this is our cholesterol molecule. All right? Maybe I should go ahead and draw the rest of that. Yeah. This is called pregnenolone. There we go. Pregnenolone, this is the precursor to all steroid hormones. All right? From here, if we want to make other hormones, we can do other tweaks. But all of them will revolve around making this a little more hydrophilic, or just changing it in some ways to allow it to be differentiated by different receptors. All right? So from here, we get converging pathways, all of them resulting in some kind of steroid. A steroid is a hormone based off of this sterile ring structure. Let's just go over a couple of them. We're not going to go over the biosynthetic pathways. We're just going to look at them. All right, so we've made them more hydrophilic. They could be signaling molecules. Here's 2 of them. The difference between me and her is this one right here. That's it. Some part early in our development, I started making this. Otherwise, I would have been a girl. Same with the rest of the guys in here. This is testosterone. That's literally it. And some other androgens are made. And then later on in development, I would have made something called anti-Mullerian hormone, something that would have prevented the formation of a uterus. Otherwise, I would have had some mix-ups going on down there. One protein hormone and testosterone. And we're very different. She's much better looking than me, for starters. This is estradiol. They don't look all that different. The receptors that bind these things, the testosterone receptor binds around here-ish, estradiol receptor binds around here-ish. Not much chemical difference between the two. Huge differences, right? All because at some point in development, I started making testosterone. The fetus is actually inundated with estradiol and other estrogens from the mother during development. So we're all fated to be girls unless we start making testosterone and unless we prevent the formation of a uterus. This is all done with very few compounds, all right? So this is one important implication of steroid hormones. The difference between boys and girls, all the differences come from the presence of testosterone early on in development. That blows my mind. Come on, that's cool. You guys got this. That's just amazing to me. They're practically the same thing. I wouldn't be able to tell the difference if I saw those on the street. I think they're pretty much the same thing, but we can tell the difference. There's another important one we're going to go over, because we kind of alluded to it before. That's the formation of vitamin D3. All right, so how that works, this is pretty cool. I know I say everything's cool, but it really is. Kind of messed up, but it's all right. Okay, this is 7-dehydroxycholesterol. Right, this is synthesized and then in the liver and then transported to the skin to undergo its next synthesis step. That is UV light will interact with this small conjugated pi system there. You can tell because it's a small conjugated pi system, only high energy light will be able to interact with it. Results in the breaking of a bond. So we get this kind of rearrangement happening. Yeah. And we break right there. Form a molecule that looks like this. Forms this molecule, which will spontaneously rearrange. This is what I summarized. Basically, these double bonds hop around. That happens spontaneously. Okay, this is vitamin D3. It's not done yet. There's other things that are going to happen to it that we won't go over. This is what's in all our milk. Right? Because we need it for a variety of things. One important thing would be how we respond to calcium transport calcium around the body. That's one important thing. It also has other roles that we're not really clear about in the immune system, things like that. This UV light step. It's the reason why people of European ancestry are so pale and prone to skin cancer. It's because we needed to do this chemistry, but we were at these high latitudes where there wasn't much light. We had to lose all our pigment in order to do this chemistry. That's why we did it. But then there was a trade-off. If we decided to move back down south, we'd get skin cancer, we'd have to get more pigment again. So as the peoples of the earth move up and down in latitude, their pigment adjusts to balance skin cancer with the production of vitamin D3. All right? Very important chemistry. All right, so that's it for steroid hormones. Let's move on to biological membranes. So, steroid hormones, they all start off as cholesterol, and then we make them more hydrophilic. All right, and they do important stuff for us. Yeah, summarize it. Okay, we're going to talk briefly about the properties of biological membranes before I let you guys out of here. Okay, so there's all kinds of different organisms that have biological membranes, all of them on the planet, in fact. And there's some common elements between these different membranes, and there's some elements that are specific to certain organisms. Let's talk about the things that are common. So here's our membrane. I'll kind of be diagramming membranes like this. We have a hydrophobic core. And then we have some polar head groups that interact with water. Polar just means hydrophilic. Okay, so membranes. One thing we've talked about with membranes is that they are all fluids. And that's important that they are fluids. Let's put a little asterisk there. When I think of fluid, I think, yes, sir?
I can't see the red pretty much at all from the glare.
Oh, I'm sorry. From up there, or
No, the bottom.
bottom one?
Yeah.
All right, well, this is just my little membrane diagram. You can draw one. I'll switch to black for the rest. So I just wrote membranes here, and that they're fluids underneath. You can't do that. All right, and then I put an asterisk next to the fluid. Because when I think of fluid, I think of something like water, where in 3 dimensions, the molecules can move past each other. The fluidity of membranes is not 3-dimensional. So let's think about a glycerophospholipid. Sitting here in this membrane. He's a fluid. He can move past his neighbors. But it's regulated. It's limited to lateral diffusion. Lateral diffusion is rapid in membranes. All right? There's another degree of freedom that these membrane lipids are not allowed, and that is the ability to flip which face of the bilayer they're on. This is not favorable. All right, on Monday we'll talk about just how not favorable it is. We'll put some numbers to that value. To this idea of not favorable. For now, just trust me, this doesn't happen, or happens very, very slowly. Rare event, but diffusing laterally happens just fine. All right, we can summarize why that is because of this polar head group. If we want to move and flip this, that polar head group has to be dragged through this hydrophobic layer. That's not a favorable reaction. Okay, so it's fluids with an asterisk. All right, we have lateral diffusion. We do not have free diffusion in 3 dimensions like we would have in water. All right? Another cool thing about membranes is approximately 50% of the mass is protein. In bacteria, that goes way up to 70 or even 80%. So in bacteria, most of the membrane is actually protein. Just some lipids holding it all together. Right? Lots and lots of biology is happening in these membranes. Some would argue most of the biology is happening in these membranes. We don't learn about a lot of that biology because it's really hard to study. If anyone's ever worked with or gotten tar on their feet at the beach, they know they don't want to be working with lipids for the rest of their lives. So we know a lot about the biology that happens in here, but not much about what's going on in these membranes, even though it's almost all protein, it's almost all biochemistry. That's what's going on there. It's not well studied. We'll talk about some of the things that have been studied next week. All right? The leaflets also, the leaflets are asymmetric. So first, what's a leaflet? This is a leaflet, just one part, one half of the bilayer. This is the other leaflet. All right, what I mean by asymmetric is there's differences between these two. So if we're talking about a eukaryotic cell, so if this was the outside of the cell, this was the inside, there are going to be differences between this and this. These differences could be in the head groups, it could be in the proteins. One important difference that you guys are probably familiar with is glycosylation in eukaryotic cells. Glycosylation events and the glycosyl groups are only presented to the outside of the cell. Right? They're not presented to the inside. So that's one way in which they're asymmetric. We'll talk about many more on Monday. All right, so the leaflets are asymmetric, and this is important because it allows the cell to tell the difference between the inside and the outside. That's important. Right? The leaflets are asymmetric. Also, membranes are selectively permeable. Selectively permeable. Some things can make it in and out of the membrane, no problem. Other things can't make it through at all. It's very energetically unfavorable. It all has to do with this hydrophobic core and whether or not an interaction with that over interactions with water are favorable. That's what determines the permeability through a lipid-only membrane. The cells can also regulate the permeability of a membrane by putting transporters or channels in the membrane to allow things to pass across. Things like glucose. That's one I'd want to get across my membranes. Okay, so these are the major features of biological membranes. We're going to go over these next week in exquisite detail. We'll be talking about what limits, I guess we don't need to talk about what limits the fluidity anymore. We'll be talking about the selective permeability first. Then we'll be talking about the asymmetry. How can we figure out what's the outside and what's the inside of a membrane? Then we'll be talking about membrane proteins and what they do and how they contribute to the properties of the membrane as well. All right, so I'll let you guys out of here a few minutes early. Go enjoy your weekend. We'll resume with membranes on Monday.